Episode of Zoology, and this is a laboratory prelude for beginners of this course. This is an introduction on how does the basic life forms have emerged and where did they originated from, and we will talk about more of the basic unit of life, which are the cells. Okay. Um, Scientists are trying to work to determine the accuracy of understanding the relationship of organisms when it requires changing established concepts. So the scientists are working with different researches because they want to try to prove what are the phylogenetic relationships or the origin of these animals. Okay, they're trying to fill in the gaps, to fill in the links that are trying to hinder their researches regarding the developmental pathways of these animals, their morphological structures, and their DNA sequences. Okay, so they are using fossil records or the remnants of these animals and plants to understand the complexity of life. Okay, so to understand the evolutionary history of life or the phylogeny and to represent this in a hierarchical classification or taxonomy, they have to work hard for understanding these concepts. In order to do that, we have new methods of generating and analyzing the evolutionary hypothesis to continue their understanding of phylogenetic relationships, because classification schemes will reflect phylogenetic relationships because these have an explanatory power. Okay, to understand an animal and where it should belong, it must have being proven by different kinds of set of data. Okay, different set of experimentation that underlies within, so that you could update, and scientists are currently updating their classification schemes to avoid misconceptions, to avoid miscommunications, and also to reflect these advances in knowledge. So in 1758, Carolus Linnaeus, a Swedish naturalist, assigned all known kinds of organisms into only two kingdoms. We have the animals and plants. But over the centuries, this dichotomy of plants and animals served different biologists, different researchers to identify the hypothesis of shared common ancestry by three major evolutionary lineage. So we'll try to remove the two kingdoms, plants and animals, because we have already discovered the DNA sequence data or of all of our hypotheses. Okay, and it corroborates numerous other lines of evidence as well. So we will abolish this one, but Carolus Linnaeus have devised a classification scheme. We call that binomial nomenclature, wherein the genus name is written on the first part with the scientific name, and then the second name with the species epithet name. So we will also thank Carl Linnaeus for that. So these are the three domains. Okay, major domains are bacteria, archaea, and eukaryota. We'll talk about the archaeans. Okay, they are living in the harsh conditions, maybe in the volcanic vents, ocean floor, into the deepest part of the ocean, into the hottest part or coldest part of the world. And for domain bacteria, these are the true bacteria, and they can be classified using a staining technique. We call that gram staining technique, and then they are classified into gram negative or gram positive bacteria. For domain eukaryota, these are the single-celled multicellular organisms, and that includes the animals. And we have here three kingdoms in one supergroup: the kingdom fungi, the supergroup Archaeoplastida, a kingdom Plantae, and lastly the kingdom Animalia. An example here in the kingdom Animalia is a Cameleo calyptratus, the veiled chameleon, which is a reptile. Also has the ability to change its colors according to its mood. So that's one characteristic of reptiles, of the chameleon, that they are able to change their patterns to avoid predators coming in. So we have the three domains, and we will try to check the characteristics. So the first one is bacteria, or the true bacteria. They are made up of a prokaryotic cell, a cell that doesn't have a nucleus, a singular chromosome, circular chromosome, a cell wall, okay, a thick cell wall containing a peptidoglycan, and they're also a chemosynthetic autotrophs. I mean to say, they are living in they can manufacture their own food using chemicals. Okay, they can also use the you can also have a chlorophyll based photosynthesis. They are also photosynthetic autotrophs that relies energy from the sun to manufacture their own food, and they are also heterotrophs. They rely on the flesh of the other organism. Okay, they have these two forms, positive and negative, and they don't have a nuclear envelope. They lack organelles and cytoskeleton. For archaeans, they're also prokaryotes, single circular chromosome. They have a cell wall. They have unique membrane lipids, ribosomes, and RNA sequences, but they lack nuclear envelope. Some of them have chlorophyll-based photosynthesis with organelle and cytoskeleton. So archaeans are more likely to have evolved such little complexity compared to bacteria. But for eukaryota, we have nuclear envelope enclosing more than one linear chromosome. Okay, so for different types of animals, we have a lot of numbers of chromosomes found. Okay, membrane-bound organelles in most, some of them with chlorophyll-based photosynthesis for the kingdom Plantae. So what are the common classification system of some groups of living eukaryotes? So we have the excavata, okay, the diplomonads, parabasalids, and eugenozoans. For chroma alveolata, the alveolates and the streminophiles, the rhizaria, archaeplastida, and uniconta. And for animals, we belong to the eukaryote supergroup Uniconta, ophistocons, the nuclearids, the fungi, the choanoflagellates, and animals. So the single-celled eukaryote supergroup phyla are consisting of a heterotrophic, or those animals that relies on other animals for food or diet, and phototrophic for those who are relying on the sun. Okay, so these are protists. So phylum Amoebozoa for amoebas and slime molds, phylum Heterocontophyta, the water molds, the atoms, the golden algae. Eugenozoa are for eugenoids. Cryptophyta for cryptomonads, phylum Rhodophyta for red algae, Dinoflagellata, dinoflagellates, phylum Haptophyta for haptophytes. These are the kingdom Animalia, and we'll start with the invertebrate for those animals that doesn't have a backbone, and vertebrate animals does have a complete set of backbone. Okay, for phylum Ctenophora, the comb jellies, phylum Porifera, the sponges, Nidaria, coral, hydra, and jellyfish, the chordata, lancelets, tunicates, and vertebrates, the phylum Echinodermata, the sea urchins and the sea stars, Hemichordata, acorn worms, Nematoda, the roundworms, Nematomorpha, horsehair worms, Tardigrada, water bears, Arthropoda, crustaceans, insects and spiders, Chinorhynchta, spiny crown worms, Bryozoa, the moss animals, the Entoprocta, the goblet worm, Annelida, the segmented worms, Mollusca, the clams, snails, and squids, Nimmertia, proboscis worms, Brachiopoda, lamb-shells, Phoronida, horseshoe worms, Gastriotricha, hairy-blacks, Platyhelminthes, the flatworms, and the Rotifera for the rotifers. So this is the phylogeny in the classification of metazoa, or the multicellular animals, as it was being read earlier. This is a tree of life, okay? So this started with this metazoan here. Bilateria for bilaterally symmetrical animals. That means to say that they have the mirror image of the left and the right side of their bodies. So we will discuss that one on the patterns of organization in the next lesson. So these are just some representatives of the kingdom Animalia. So let's just focus more on some of these representative. Okay, let's talk about the mollusca. Okay, the clams, snails, and squids. So take a look at this one. They have bilateral symmetrical with a true coelom. They have containing a mantle. Okay, the mantle, and they have a muscular foot and protective shell. So for the snails, try to look at Gary's foot. Okay, it has muscular muscular features, and it's also protected with a shell. For arthropoda, they have segmented bodies and paired and jointed appendages. So the appendages are are six appendage. Okay, but for some arthropods like the spiders, they have eight. Okay, they have chitinous exoskeleton. The exoskeleton is a outer skeleton, so they are made up of a protein named chitin, and the hemocoel for blood flow. So these are the structure of your cells. Okay, so we'll take a look at the components of your cell, the structure, and the function. So for cell membrane, they are composed of a protein phospholipid molecules, and they control what comes in and out of the cell. It also forms the cell. Cytoplasm is a fluid gel-like substance. The cytosol makes it fluid-like, and it suspends the medium for all the organelles and dissolved molecules. For the ER, it's interconnected membrane-lined channels. It enables the transport of metabolic chemicals. For ribosomes, it is a granular of nucleic acid. It creates protein. For mitochondria, it's a double membrane sac, cristae, and chambers. It assembles the ATP or adenosine triphosphate for cell respiration. For Golgi complex, it has a flattened membrane-like chambers. Okay, it's a stacked. It creates and transports molecules for secretion. For lysosome, they are also connected with the Golgi complex. They are surrounded with enzymes. It digests foreign molecules in worm cells or the waste products. Centrosome are a mass of protein that may contain a rod-like centrioles. These spindle fibers also assist in mitosis and meiosis. From vacuole, these are membranous sac and secrete substances. Okay, it allows the movement of bulk materials coming in and going out of the cell. It regulates the cellular turgor pressure. The microfibril and microtubule is producing the protein strands and tubes, which is the part of a cytoskeleton that makes up the cell or the framework of your cell. Cilium and flagellum are the cytoplasmic extensions of a cell containing the microtubules. It helps the locomotion and it propels the animal cell going elsewhere. The nucleus is the most complicated part because it contains the chromatin or the DNA, and it is surrounded by a nuclear envelope, the nucleolus, and it contains genetic code to direct the activity of your cell, and it also forms the ribosomes. So this is a prokaryotic cell, as it was discussed earlier, the domain bacteria and domain archaea. So this is a generalized cell, letter A. For letter B, this is a bacterial cell. So it is a long bacterial flagellum that helps it to move. It does have a DNA, but they are circular, and they are floating in the cytoplasm of your prokaryotic cell. Whereas in a eukaryotic cell, they are compartmentalized and they are housed inside your nucleolus. It's surrounded by the nuclear envelope, going out in the nuclear pores, passing through the endoplasmic reticulum, going out in with a vesicle and transferred into a Golgi bodies, and transferred into a vesicle again, going out in the cell membrane, and it will be released outside. So the simple machineries of your cell, of a typical animal cell, is very complex. So another study we call cell biology or molecular biology to study the mechanisms that happens inside the cell. So these are just the parts of your cell. Okay. So what is microscopy? So in order for us to view the cells, we need microscopes. These are the apparatus that is being used in the laboratory for us to magnify and to resolve the images that are found in the textbook. Okay, to view the different subcellular components of your cell. So this is a compound monocular microscope for letter A, and this is a compound binocular microscope for letter B. This has different parts. Let's start with the eyepiece, the head, the arm, the nose piece, the objectives. The objectives have the scanning objective, the low power objective, high power objective, and the oil immersion objective. So you need the oil for the light not to scatter on your stage mounted slides. Okay, so number 6 is a stage clip. It clips the slide. Coarse focus adjustment knob for big leaps of movement going up and down. The stage is number 8. Number 9 is the condenser. So the condenser regulates the amount of light coming in of the stage. Okay. 10 is fine focus for minimal adjustments or minimal movement going into the objective of your microscope. We have the collector lens with iris. Okay, you regulate the light there, you adjust the light, and the illuminator inside, the light, and the base. So always have to take care of this microscope, because the compound light microscope uses the light, and some of them are using the mirror to gather the light from the outside source, and some of them are plugged into an electricity like this one. So these are just a micrograph of it is not using the light microscope, but rather another type of microscope. We call it electron microscope. It uses electron particles that is found on the surface of your atom, and it is reflected on these images. Okay, so this is a fractured nuclear envelope showing the nuclear pores, as we said earlier. These are found in your nuclear envelope. They have protein complexes that allows the transport of water-soluble molecules into the nucleus. If they are found in their cell membrane, they are also called aquaporins. Number figure 1.5, an electron micrograph of a centriole. This one, letter number 1, and number 2 is a centriole in the longitudinal section. So these are also an electron micrograph of the lysosomes. We have nucleus in number 1 and the lysosomes in this dark stained circles, the little here. And number 1.7, an electron micrograph of a mitochondrion in a cross section. It has an outer membrane, an inner membrane, and the crista. For this example, the 1.8, this is also an electron micrograph of a cilium in transverse section, and they have this 9+2 arrangement of macrotubules that helps in the aid helping the cell's framework or the shape of the cell. Okay, the cilium is also helping in the movement, microvillus in an electron micrograph. This is a smooth endoplasmic reticulum without the ribosomes attached. An electron micrograph of your rough endoplasmic reticulum. Number 1 is the ribosomes, and number 2 is their cisternae. And number 3 is the RER, containing collagenous filaments to the outside of the cell. So number 1 is nucleus, number 2 is RER, the 3 collagenous filaments, and number 4 the cell membrane. So this is an example of an epithelial cell from a cheek scraping. So in the laboratory, you will be doing this. The actual laboratory will let you scrape from your cheek, okay, using a toothpick, and then submerge it in using a dye, okay, stain it, and then place it on a slide, and then cover slip, and then try to view it under the microscope. So we will see the 3 basic parts of your eukaryotic cell. We have the nucleus, the cytoplasm, and the cell membrane. This is an erythrocyte, a blood cell, in a 1000x electron micrograph. So it has a disc shaped. Okay, this is in an isotonic solution. If you learned it from your laboratory, the movement of water, okay, has an equal proportions. So these are the leukocytes. Okay, the white blood cells. Note that each photo contains several erythrocytes, or this one, the red one, or the red blood cells. And these red blood cells don't have a nucleus. Number 1, neutrophil, basophil, eosinophil, lymphocyte, and the monocyte. This is a capillary containing an erythrocyte. Okay, number 1 is a lumen of the capillary. Number 2 is the erythrocyte with the disc shape. Number 3 is an endothelial cell, and number 4 is the nucleus of your endothelial cell. This one is an micrograph of a skeletal muscle, a type of tissue. Okay, a myofibril showing the striations. Number 1, mitochondria. Number 2 is Z-line, A-band, I-band, T-tubule, the sarcoplasmic reticulum, the M-line, and the sarcomere, which is the basic functional unit of a myofibril. Number 1.18 is an osteocyte, another type of a connective tissue, a bone cell, in a cortical bone matrix. Number 1 is a lacuna. Number 2 is the nucleus of your osteocyte. The 3 is the bone matrix, and number 4 is the canaliculi. And lastly, we have the 1.19, a neuron smear, another type of a tissue, a nerve tissue. Okay, number 1 is a nuclei of surrounding neuroglial cells. The neuroglia is the one which nourishes which nourishes the neuron. Okay, nucleus of a neuron in number 2, nucleolus of a neuron number 3, and the dendrites of the neuron, or the tree-like structures of your nervous tissue. So that's it. We have discussed the cells, the basic of the some of the tissues. We will be um, getting more in that in the from the previous video that I had in cells, tissues, organs, and organ systems for the lecture. But for the laboratory, we'll just focus on these electron micrographs for you to be able to see what's the actual photos or actual materials found. Thank you so much for listening.